Hello, physical chemists. I am now going to answer your questions, and I'm going to put it on YouTube in case anybody else has those questions and wants to wants to listen. So we are now 3/4 of the way through the book. We're going to finish up the book pretty fast. It's actually going to be in a week. We're going to have more of the questions like this. But actually, I'm really enjoying it. Usually this year I ask the questions in person. Obviously, we can't do that. I was thinking about doing it live, but also with schedules and things like that. I figure asynchronous is the way to go. And your questions have given me the chance to really sit back and think and actually research some of these. I've had to cut myself off from researching them too much, but I do want to thank you for your questions, and I want to answer them as best as I can right here. So, going through the 6 questions that we have in order from chapters 7 through 9. First question, would plants have a different reaction for photosynthesis if they were a different color? And by different reaction, it depends on what you mean, because the reaction has a lot of parts to it. The fundamental reaction for photosynthesis is going to be the same for plants, because it depends on having an abundant source of light and an abundant source of electrons, which is water in this case. Once you figure out how to use the light to pull the electrons off water, you have a very abundant source of electrons. It's a pain to pull them off, right? But it's not the kind of thing that you would have any reason to move away from as long as you have enough light and enough of the manganese and other things that you need, but usually you need small amounts of that. Once you have enough light, you would actually have enough enough materials. So that's the reason why plants go through so much trouble to pull electrons off of water, because once they figure that out, and because light is powerful enough to pull them out, once they figure that out, they're able to basically have an endless supply of electrons. It's almost infinite, because water is pretty much everywhere. So the thing is, would plants have a different reaction if they were a different color? They wouldn't have a different fundamental reaction. But the reaction involves absorbing the light as well, and you would have to have different machinery for absorbing the light. So in the sense of absorbing the light, I would say that there is a different machinery that you would have to have, different proteins and different molecules. Chlorophyll itself is modified slightly to absorb different wavelengths depending on the light that is available to the plant. So it's not a different fundamental reaction, but it's a different molecules, the different shapes of molecules to absorb the different shape of light. Light that's a different color is actually a different shape if you think about the wavelength as being its shape. The light harvesting complex as a whole will be different. And it's very interesting to look at what those differences are and how they work. It'll be tuned to absorb the most possible light. Also, one thing that I think about overall is that some colors don't provide as much energy. Red light has less energy than blue light. Now, I'm not sure how significant this effect is, but I can say that it would be a different energy from the reaction, and you would get a different quantity of energy. The quality of the reaction would probably be the same, but I think the quantity could be very different. So, a very interesting question to think about. And so plants actually change how they collect the light and how much they process the light, as in what proteins do they bring out to run the same fundamental reaction. And so I found this article about how plants wait for the lights to change to red, okay? They actually have to do a balance between the cost it takes to build the proteins and harvest the light and the energy they get back from that light. And reading this right here, they're going to, and they actually do care about the quality of the light. In fact, they will detect red and far red light, and so they will actually allocate their resources differently in the different colors. So not a different underlying reaction, but a different transcriptome, a different level of proteins, all sorts of differences in the plant itself. Chapter 7, I love to talk about this stuff because you asked a quantum biology question, and what better place to talk about that than in a physical chemistry class? How do the pigments manage to sync their quantum states? I said that it did really briefly, but of course I didn't give you the equations. I don't understand them myself, but I'm going from articles like this, and this is actually a good high-level sort of explanation. that doesn't get too much into the details if you want to look at this about how quantum mechanics actually affects biology. Part of that has to do with quantum biology in photosynthesis. That's what I'll talk about. It is related to orbital hybridization because it has to do with how the electrons move, and of course the shapes of the orbitals are going to affect how the electrons move and how different things overlap. I can't go into too much more detail, but I will say yes. I'll answer your question and say yes, it is related. Here's a figure that shows what the quantum biology of photosynthesis is. And here it's about how quantum biology, an electron, because it has quantum mechanical behavior, can actually sample all possible paths and choose the best one. Remember we talked about that in refraction, actually. There's a similar thing that goes on in photosynthesis. In the sense of refraction, we talked about the path that the light took when it went through an air-water interface. With photosynthesis, it's the path that it takes from where the photon comes in, excites the electron. The electron then has to travel to the reaction center, shown in orange on this. And it turns out that the electron actually takes the most efficient path, just like light takes the most efficient path through the air-water interface. And so there actually are a lot of similarities to it. And the reason why it works is because, in a sense, the electron takes all possible paths. The traditional model is that the electron would hop around, but this would result in a lot of wasted motion. The quantum model, see right here, the electron is taking all possible paths, but then because of the nature of quantum mechanics, it's able to sample all those paths, and the one that persists is actually going to be the shortest path. You see right here, it's the shortest path, 1, 2, 3 spots to get from where the photon comes in on the left to the orange reaction center on the right. And so they've done tests on this, similar to, you have to test how the electrons move. So it's almost a physics experiment that you're doing on the plant photocenters. It's very cool. I don't understand it all, but I do know that it has to do with the way that light and electrons behave. It's much more possible for an electron, which is very light and has quantum mechanical properties that are more evident, than for even a proton, or definitely more than that. By the way, there's similar kind of quantum superposition things that do happen with protons. Once you get above a proton, there's really less and less of it that you can see. So I'm doubting that it will become much more important as you get above electrons and protons. But it's pretty important right here. Another question from Chapter 7. A lot of people want to talk about plants. A question also about photosynthesis. Plants get their electrons from water. Do plants obtain any source of electrons from the soil? I can't imagine that they obtain many electrons from the soil, but the way you phrased the question was not, do they obtain many electrons from the soil? Do they obtain any electrons from the soil? And so this got me thinking, and so this is my best answer to it. We know that they accumulate metals, and then they can redox transform those metals. But they appear to do this in response to the metals being toxic, the metals acting as metals, and they don't appear to harvest electrons from it. Again, once you can pull electrons off of water, all you need is an abundant source of light, and then you have an abundant source of electrons. My question would be, in low light situations, could there possibly be some plants that are able to pull electrons off of metals? And we know they can transform metals. I tried to look up whether they oxidize or reduce, but I think they can do either. I'm not sure. I would guess that they oxidize more than they reduce, which would mean that they actually are getting electrons off of the metals. But I'm really not sure if there's enough of that to be a significant source of energy, which gets to your question. So my answer is probably technically they may be able to do that, but I can't imagine that it would add up for them to be able to do it at a wide level. Then I got to thinking about the other forms of photosynthesis, and we talked a little bit about iron photosynthesis, and in the book it talks a little bit about hydrogen sulfide photosynthesis as a possible stepping stone. Bacteria can still do these different kinds of photosynthesis, and so maybe the plant can't do it, but maybe there are some symbiotic bacteria that can do it and then pass it to the plant. As it adds up, I think that if it happens, it's kind of a curiosity, rather than something that really affects how energy moves through the biosphere. But I think that symbiotic bacteria, we know they can do these other kinds of photosynthesis. The plant can get some benefit. Maybe the benefit won't be as far as electrons that it gets, but it could get nutrients, and for example, like a plant could get nitrogen, usable fixed nitrogen from a symbiotic bacterium. We know it can do that. So maybe there's some kind of thing where the bacteria are doing a different kind of photosynthesis. In that case, it would be the bacteria doing it, really not the plant. I would love to be surprised on this point, but I'm not sure how the energetics would hang out. So I'm going to make my prediction that no, I don't think this happens that much, but it's a very interesting thing to think about and think through. Here's a nice geology question that we have when we get to Chapter 8, which actually has a lot to do with geology. How do scientists find out what the oxygen levels in the atmosphere were 1 billion years ago? So, as you know, my book is already long and detailed enough without me putting in the exact geological methods for how scientists determine these things. And they are somewhat contentious because it's not like you can exactly just measure oxygen with a little oxygen probe that looks back 1 billion years. You have to be looking at the geological elements, which means the redox-sensitive metals that are in the rocks. You're never looking at oxygen directly, except in the weird case where you have inclusions in the rock, and even those have complications. So you're always looking indirectly, and it's complicated and involves a lot of things about isotopes and the redox state, whether you're seeing, iron 3 or iron 2 is a simple example. But this gives me the chance to show you some data where scientists did this, and this just came out. It's actually really cool and affects the world from dust story. Remember how I talked about how there was an oxygen spike early on, and it looked like that spike was very narrow, like 100 million years. And I know that's not really narrow, but relative to Earth history, relative to 4 billion years, 100 million years is pretty darn narrow. Well, we found more evidence that that spike continued. That evidence was found because they found high oxygen evidence in rocks that were 2.1 billion years old. Well, now they found that evidence up to 2.0 billion years old. And that was actually a pretty big deal because it inspires some thinking about, we see that the oxygen drove so many things. Could oxygen have driven a eukaryotic evolution at this point, and then oxygen pulled back before it could drive multicellular evolution, and then we had the boring billion? Very interesting possibility. The wider the early spike, the more possible it is, in my view, that oxygen could have driven this very important early event of the mitochondria and endosymbiosis and all that fun stuff. And so, and they actually say as much. This highlights the importance of realizing we could have had a lot of oxygen. The important thing is we still know this is the width of the spike, and the question is did it end at 2.1 or 2.0 billion years ago? Realize how narrow that question is. But I actually like this, and I want to show you what their data were. Here it is. This is, I think you can zoom in, or you can pause and zoom in at the very top of these. Look at the top and bottom of this figure. The bottom of this figure, you see the different colored boxes, and those are different kinds of stone. This is how geologists show their data. And of course, they're always getting core samples, so they're always digging down into the earth. And you can see the depth of these samples and it's actually literally, 1 kilometer down is where they found some of these depth meters, 1,060 at the top. So they're literally taking a long chunk of meters of rock. They show you the different types of rock stacked up on each other as the colored boxes under lithology. And if you look to the side, for each of those boxes, they are measuring different chemical characteristics. Geology is very closely related to chemistry. The main thing I want to show is this is all chemistry when you look at it. You could argue that it's physics because it has to do with isotopes as well, but I'm going to call it chemistry because I see elements there. You're going to see molybdenum and uranium right there. Both of those are, molybdenum at least is redox sensitive, and so it is going to be different, and the amount of molybdenum that goes into the rock will be slightly different depending on the level of oxygen and the isotopes of molybdenum. And so they can look at the isotopes. If you look over on the left side, you see a little delta 98 molybdenum. It's hard for me to see, but that's a sign of an isotope. You see 238 uranium all the way on the right in the farthest right column. And I even see, I believe that's rhenium. I'll have to look at the thing to see. Molybdenum, no, it's actually probably something else. It doesn't say what it is. It might be rhenium, an element. Actually, I think it is. I know it's molybdenum, and again, we've talked about how important molybdenum is for understanding what's going on with oxygen. I believe that's where they're pulling a lot of this from. Uranium is also oxygen sensitive. Remember, we talked about on the oxygen-free Earth, you had uranium pebbles that would oxidize away very quickly in an oxidized environment. So there's a lot of evidence for the times when there wasn't oxygen around. They're finding more evidence to find out where and for how long oxygen was around during this weird spike that was 2 billion years ago. But this is the evidence, and that evidence has gotten wider, and since I think oxygen is important, I think this kind of evidence is really important, but I'm not in a position to critique it, and of course you aren't either. So we'll just take the geologist's word for it, let them fight it out to figure out how long oxygen was around. But I like this study because it goes with my prior conclusions, right? Here's another study, Chapter 8, and this almost got me started on a rabbit trail because the answer is yes. Basically, the student was looking at zinc and calcium and saying, well, all your reasoning for calcium looks like it would actually work for zinc as well. Wouldn't zinc be a possible idea to work like calcium works? And you've actually hit the nail on the head to find an entire research area, because it looks like zinc is not quite as widespread as calcium, but it's a clear second place, and it's even used for synaptic signaling, exactly like what you're saying right here. In neurons, zinc is used as a sort of neurotransmitter, so it's sort of halfway between being like calcium and being like glutamate, okay? In fact, it's probably used a lot like glutamate, but the way it works, you can see that it's actually, it goes as zinc 2 as soluble zinc, and it does cross from cell to cell. So the example is, if you can do a good chemical reasoning, you can look for evidence of whether that actually happened. I think zinc is one of the best examples of that. Now, there's a scientist, what the rabbit trail was for me, as I'm remembering, and you'll have to go from my memory because I could not find the evidence for this, but my memory is very clear on this. A very prominent scientist, and I believe his name was Stephen Lippard, but I couldn't find this in a quick search. But he was getting up and he was talking about like he's sort of at the end of his career, and he's saying, what would I study if I was starting my career right now? And you know what he said? I would study zinc in neurons because it's so chemically cogent. It's such a good idea, and it's so clearly important from my study of it. So there you go. And maybe you can think about studying zinc in neurons someday. I don't know if, but that's what he said, and it really stood out to me, because here's a scientist saying, what would I do today knowing what I know now if I was starting over? And this shows me in general, this chemical reasoning works. It gives you a place to look that pays off in the terms of research. So this question you were able to formulate was actually the basis of an entire research area that is taking off right now, because we have found evidence that this indeed happens. Okay, so I was excited to find that, but of course I'm like now, okay, I want to see the evidence, and then I had to cut myself off. I'm going to have to cut myself off from this as well, because we have, I believe, one more question, Chapter 9 question, about the speed of evolution. We talked about examples of speeding up evolution under stress, freezing evolution in the absence of stress for plants. Could the same be said about animals? And yes, it's hard to prove, especially with animals. You can definitely say it with bacteria. You can even run the experiments with bacteria. Plants, I can say it pretty confidently. Animals, probably. And the best example I can give right here is, have you heard of and I hope I'm saying this right, the coelacanth, which was supposed to be a fossil, and then they found it swimming around in the Indian Ocean. Some fishermen pulled it up in their net, actually. I remember growing up and finding out that it was being told it was extinct. And then I saw that some fisherman pulled it up in his net. And so the coelacanth does actually exist, and we found out where it lives now, and we see it living still today, and there's a diver right next to it. So we had this in fossils like 300 million years ago. Then we didn't have any fossils, and we thought it was all gone. And then, but it's not all gone. And so this is a really interesting example. Do we have evidence that this genome is evolving more slowly? And the answer is yes. Back in 2013, the coelacanth was actually sequenced for the genome, and they compared its protein substitutions, the substitutions in the amino acids, for the different sites in the protein to other related fish, lungfish, chicken, and mammals. And mammals have a 1.2 rate of substitutions, lungfish and chicken about 1, and the coelacanth has the lowest, 0.9. So it's not like glacial, it's not like it's not happening at all. You can't keep mutations from happening. But from this, people were saying, it sure looks like it's slower. Is this a cause or is this an effect? How is this done? Those questions are still open. But the substitution rate is the easiest thing to look at, and it definitely is slower. Down here they say the reason for the lower substitution rate is unknown, although they refer to the exact same things that the student asked this question did. A static habitat and lack of predation could be contributing factors to a lower need for adaptation. It definitely fits. Now, I've tried to look to see if people have looked into this a little bit more, and all I found was this particular thing right here. There's the Hox genes, and ask Dr. Tenlin about these, because they are related to development, which is her area. And but it looks like there's some things about the Hox genes that are kind of slow. They're arranged so that development doesn't happen as quickly. I'm not quite clear about it because I haven't gotten into it. But they've looked at the arrangement of Hox genes, and they suggest that that might have an effect on the rate of evolution. We know that the coelacanth has not had significant morphological evolution, because we've seen that on its, we can look at it, and we can say it looks like the fossil. But on the other hand, the coelacanth, what does that mean for its genes? We got evidence that its substitution rate is slower, and is that because it has, in a static environment, the genes that are rearranged to have slower rates of substitution somehow have conferred a selective advantage and allowed it to survive more? The genes are changing slower. The shape definitely hardly changes at all. The question now is why, and what mechanisms cause it to do this. But this is strong circumstantial evidence that the reasoning is appropriate. If you see something that its shape is the same, it seems to be valid to say that, in a sense, evolution has slowed down for it. Is it because the environment has a very slow change? Is it because that the lack of predation doesn't drive change there as much? Those seem like the most possible possibilities, but once we get to this level of question, it's the one where I want to get you to the level where you're asking these exact questions, because I can't answer them. But I can tell you, you'll be able to keep on asking these questions for years and years, and maybe they'll be answered better and better as we go along. Maybe something about how the Hox genes are arranged. Very interesting to look at that. All right, thanks for all your questions. Now get to work on all the other stuff that I've assigned you, including the last 3 chapters. I look forward to your questions on those. I'm going to keep doing that because I enjoy this, and I think it's a good way to do this class. So I look forward to your questions on the last couple chapters, and if we get outside of science for those, that would be okay too. Ask whatever questions occur to you, and I will try to look up answers. Challenge me. It'll be fun. All right. Have a good weekend.